Let's see if we are live. Let's see if we are live. I think we are live now.
Okay, so hello everyone.
Right, so let me introduce Noor. It's our great pleasure to have Noor today to present about genomic data privacy.
So hello everyone.
Right, so let me introduce Noor. It's our great pleasure to have Noor today to present about genomic data privacy.
So hello everyone.
Right, so let me introduce Noor. It's our great pleasure to have Noor today to present about genomic data privacy.
So
So let me introduce Noor. It's our great pleasure to have Noor today to present about genomic data privacy. So let me introduce Noor. It's our great pleasure to have Noor today.
Mohammed, can you try now, please?
Right. So yeah, it's our great pleasure to have Noor presenting about the genomic data privacy. This is the 10th lecture. Probably it will be our last or second last lecture. We may have one more next week. And yeah, good luck, Noor.
Yeah, thank you so much, Mohammed. Hello, everyone. So today we will have our 10th lecture, and I'm gonna explain about sharing genomic data in a privacy-preserving way using a widely used mechanism which called differential privacy. In today's lecture, we will go for why we need to share genomic data, what are the privacy risks we have, can we apply differential privacy to share the genomic data, what are the limitations, how can mitigate this, and so before I try to introduce like genomic data sharing or differential privacy, let me refresh your information and give you some small information about genome. I'm sure you all are familiar with this, so I'm briefly summarizing some background information about genomic data. So inside each and every one of your cells, there's a code of instructions for your life. We can consider it as the ingredients lesson. It's the recipe for making every living things. And these recipes are uniquely yours. It's invisible signature. These recipes are written in 4-letter languages, which are A, C, G, and T. And the genome actually is the fancy word for the entire DNA. Now, like in the previous lecture, my colleagues explain a lot about genome sequencing. And you know that genome sequencing simply means revealing and putting in order your A, C, G, and T. Okay, so if the genome is the instruction manual for you, so your genes will be the individual chapters. And inside each chapter, we have codes for making eyes, for making heart. And in fact, we are 99.9% genetically identical across all humans. We are different. We have 0.1% genetic variation between us, makes us unique. Unique in color of eyes, unique in our response to medication, unique in our predisposition to disease, and so on. Now, you inherit 50% from your mom and 50% from your dad. So you have two different nucleotides. We call these nucleotide alleles. And alleles are observed for each single nucleotide polymorphism, which is the variations you have, the 0.1% variations you have. So these alleles are different forms of the same gene. Again, one allele inherited from the father and the other allele inherited from the mother. Each observed nucleotide can be minor allele, which like we represented here by B, small letter b, or major allele. Here, the capital B represents the major allele. Now, the minor allele usually is the rare nucleotide we have. Okay, according to the inheritance you get or the values of alleles in your father and mother, you may have homozygous major if you inherit two capital or two major alleles. And in that case, we give the value 0. If you inherit both, like the major and minor, then we call it heterozygous and we give it the value of 1. Now, if both are minors, we give the value of 2 and we call it homozygous minor. Okay, what are the probabilities of having these values? So this is what we call Mendel's law. In Mendel's law, you have the father, you have the mother, you have like the nucleotide, the alleles they have, and for each one, like what's the probability for you to have, for example, 0 value, 1 value, or 2 value? Let's pick one example. For example, is if your father have the value 2, which is homozygous minor, and your mother have the value of 0, which is homozygous major, then you are 100% inheriting the heterozygous. You will have the value of 1 for this SNP, and so on. Now, again, this also the Mendel law, if we have the child and the mother, so what are the probabilities for the values for the father of that child, and so on. Now, you know that, okay, the genome holds the promise to help protect your health. And actually, the genetic factors are known to play a role in 9 of 10, for example, in the United States, leading cause of death. So we need to access the genetic information as a part of healthcare, okay? So as all of you know, we are currently in the genomic era, and thanks to the fast-paced robot sequencing technologies, which you already knew about it. So these technologies result in large-scale data sets, in large biobanks we have. Currently, we have about 2.5 million sequenced genomes around the world. This number also projected to reach 105 million sequenced human genome in the next 5 years, especially after the COVID-19 pandemic, where many countries decide to study the genome data, the genomic data in a population scale. They want to leverage the power of genome. So now we have these sequencing technologies. We have from the government's project to study the genomic data in population scale. What about the population? What about the people? Are they willing to donate? So in several studies, they report the attitude of public and different countries, including the States, Sweden, Japan, and Singapore, towards the donation of their genomic data. About 70 to 90% of these participants, they had positive attitudes towards genomic research and donating, but they also they want to donate, but they have some issues, of course. So now what's the problem here? We have the government projects, we have the sequencing technologies, and we have the people who want to donate. The problem here actually is social, not technical. We are talking about the privacy risks. Once these data, the genomic data, if it is on the internet, of course, it will be not private. So what are the problems if the owner of one genome is identified? First, this information is financially valuable. The employers, insurance companies can exploit it against that individual with a known genome record, of course. The other big issue is that once the genetic record of any individual is leaked, it's leaked for good. It's not your like mobile password. So this will lead to important privacy concerns. It's not only yours, it's not only one individual privacy. We are talking about the relatives, about the family privacy. All will be at risk. So because of that, like we have several genomic studies which considering not sharing the whole data. We are not sharing the whole genetic record, for example. One of these widely like used or very important studies, which we call genome-wide association study. And I think you're also familiar with these studies, like Mohammed mentioned about, explain more about it in the first lecture, but quickly, like what are these studies here to, like these studies focusing on finding the positions in the human genome that is statistically correlated with a phenotype. Like for example, which SNP is correlated with disease disorder like Alzheimer, which SNP is correlated with a phenotype like height. So these are the goals of GWAS. And GWAS, it's is a case control study. We have cases who are like having the phenotype of interest, again, like Alzheimer, and controls who are healthy, for example, or disease-free. So we are trying to find the association between the SNP according to the value of the genotype for these two groups. Here, for example, we have the tables to consider the value of each genotype for each group and so on. What are the results of these data, of these studies? We have like the minor allele frequency, we have the p-value, we have chi-square. So we are talking all about the statistics. We are not revealing the genomic data itself. And according to this statistic, we can find, okay, if there is correlation, if there is association between one SNP and one phenotype. Now, like, since we don't reveal anything, we are not showing any genetic data, we only have statistics from these studies. So these studies are considered safe. Okay, it's safe until one attack happened. So we have Homer attack happened, and this attack basically like tried to reveal the membership information. Like from this attack, you are using the statistic from GWAS. And from this statistic, you could know if one individual or one participant is in the data set and if he is in the case groups. So if you are talking about, for example, very sensitive data, if you are talking, for example, about hepatitis C data set or GWAS or cancer or whatever sensitive phenotypes we have. So once you get to know that this participant is in the case group, so, okay, you reveal his information, you reveal his sensitive data, you could know that, okay, he has this. So in order to stop that, what happens after the Homer attack, a lot of these studies, like the GWAS studies, stays outside the reach of researchers due to privacy concerns. And yeah, the NIH restricts the access to the key results of the GWAS. All right, so for the hope of sharing this genomic data, we want to gain better understanding of the human genetics. We need this data. What's the solution here? A lot of studies propose different techniques for sharing the genomic data. We want to try to find a good way in order to preserve the privacy. We want the privacy guarantees. And in the same way, we want accuracy as well. So we want a balance. So we have different techniques. What we want to like explain, what we focus on this in our studies is the mechanism of differential privacy. So we can use differential privacy. We can use these mechanisms for computing over the genomic dataset or sharing the genomic dataset. So today we are going to go for privacy preserving sharing of biomedical or genomic data sets. So what's differential privacy? Like simply differential privacy, like we achieve differential privacy when the adversary or attack is not able to distinguish that the answer he got or the answer produced by an algorithm based on the data of two, like we have two data sets. That adversary cannot distinguish if the answer is coming from the first data set or the second data set. In more technical, like if we have two neighboring data sets here, one we call T and T bar, and these two neighboring data sets differ in one tuple, one row. So they are the same except for one row. So if we are using mechanism A, all right, for these data set to produce an answer, this will be like the difference between the probability of applying this algorithm will be differing by a bounded factor e to the power epsilon. Now, these data sets, again, they are neighboring, they can be like the same size, and they are different in one row. They can be, they are the same size and the content of one row is different, or they can be with different size. One of them is one row, like has one row more than the other data set. Okay, so again, here, the probability of applying algorithm A over the first dataset T and the probability of applying, again, the same algorithm over the second one, the second dataset, the difference between these two probabilities will be differ by a bounded multiplicative factor e to the power epsilon. Now, what's epsilon here? Epsilon, what we call privacy budget. And according to that epsilon value, we can consider how much privacy we need and how much utility we need. Okay, if you want more privacy, it means that you will have less utility, less accuracy. And in that case, you need to choose small epsilon value. If you want more accuracy, less privacy, then you can go for high epsilon value. And yeah, here, the bigger the epsilon value, the less privacy budget, the privacy guarantees we will have. Okay, so how to achieve differential privacy? There are different general approaches. One of the most common for a numerical dataset, since here in our statistical dataset in our GWAS, we are using like numbers. We consider the genotypes, as I mentioned, as 0, 1, or 2. So we need a mechanism for numerical dataset. One of the most common is the Laplace perturbation mechanism. So here we consider a global differential privacy in which any researcher, potentially attacker, adversary can send a query over this dataset. Then by applying differential privacy, what we are releasing? We are releasing the query result, okay? And we are adding some noise. This noise we derive from the Laplace perturbation mechanism, according to something we call the global sensitivity of the query. It's the maximum change. For example, if we have some query, so if we apply for T and we apply the sum for T bar data sets, what is the maximum change in sum? So that's what we call the global sensitivity of the query. According to that, we can calculate the noise, the added noise, and then we can add that noise to the query and just release it. And that way, we can say that, okay, we achieve differential privacy. We are releasing the query result in a privacy-preserving way. Any questions so far? Okay. So the differential privacy are used in many companies, like, for example, the crowdsourcing data in Google, Apple. We have Microsoft Research also. They are collecting their data based on differential privacy techniques. One of the census companies in the States also are using differential privacy. Can it be used for genomic data? Of course, and in many applications for location, for example, and so on. So can we directly use it for genomic dataset? A lot of studies that try to find the sensitivity to apply the differential privacy directly to the genomic dataset to reveal the query result of sum, minor allele frequency, chi-square, p-value, and so on. But there are two limitations with the current studies. What are these limitations? Okay, the differential privacy itself assumes that all the rows inside the data set are independent. Okay, we don't have any correlation, any relationship between these tuples or these rows. But this is not the case in the genomic dataset because a lot of GWAS, a lot of statistical genomic dataset are considering families inside it to study, for example, the effect of having families for this disease, if it is like how some steps can be inherited and so on. So it's a fact that we have families inside the data sets. So just applying the differential privacy with the assumption that we don't have correlation between the data set tuples degrade the privacy guarantees of differential privacy. Okay, so the people are trying to mitigate this risk, are trying to find solution, are trying to consider the correlation between the dataset rows, the tuples. So, okay, we have differentially privacy-based mechanisms. They consider the correlation, but they provide poor accuracy. They are adding too much noise to cover this gap. So adding too much noise will not help us like to have a good utility, to have a trade-off between both of them in a good way, in a suitable way. So let's go for some related work, which are considering different applications, like again, mostly like locations, like temporal data, and so on. So the first framework to criticize the independent tuples assumption of differential privacy is called Puffer-Fish framework, and in this framework, they analyze the differential privacy. They show that the differential privacy assume the independent tuples inside the data set, but they don't have any perturbation algorithms. Following this framework, we have a subclass of this work which called Blowfish framework. They are trying to add some perturbation mechanism, but they make it deterministic. They have some constraints for the adversary. Again, we have Bayesian, the differential privacy. It also uses a modification of Puffer-Fish, but they are trying also to do it for any data set which can be modeled by Gaussian Markov random fields. Here we care about like statistical genomic data set. Following by them, we have different also studies trying to do also to fill this gap, to mitigate the risk of having dependency, but some of them are adding more noise, excessive amount of noise, which we don't need. Some of them, like the dependent DP, like in 2016 for a location application, they consider the pairwise correlation. And it's not the case in genomic data, since in genomic data, we have joint correlation. For example, if you have the father, mother, and son, three of them have to be like, we have to study the correlation between three of them, not pairwise, one with another one by one relationship. And so what are our contributions regarding this? So we are trying to do two things. First of all, we need to discover the vulnerabilities of applying differential privacy directly to the genomic data set, because most of the other current state-of-the-art work are trying to do it for different applications. So here we want to show, if we have a GWAS or any statistical genomic data set, how can we perform attacks over it? So we started with attribute inference attack. So in this attack, we are trying to infer the genomic data itself. Like imagine I have zero information about one target. I know that the family are inside the data set. I want to infer the genomic record of that target only using the query result, the differentially private query result. So first we started the sum query, and then we try it with more complicated, more real world queries, which are the MEF, the MAF, and the chi-square. Also, we perform membership inference attacks. We want to try to find if that target is a member in the case group of the GWAS. Also here we use the MAFI query for that. And then we want to mitigate these inference attacks. We have to find the solution. So we contribute in finding two solutions. The first one is we are trying to add the noise. We are trying to strengthen the privacy guarantees, okay, because of the correlation between tuples we have. So we are now changing the epsilon, the privacy budget we have, according to or based on the number of family members are participating in the data set. The other way we try to do is, okay, we can still use the differential privacy, the general way of the differential privacy mechanism, but we will hide some snips, some information from the family before applying our differential privacy without increasing the noise or playing with the privacy budget itself. We are just following the standard differential privacy. This technique, we call it selective hiding mechanism. So what are the data sets we use for our attacks and mitigation techniques? We commonly use the 1,000 genomes project, the phase 3 data set. Here we have data set for 2,500 plus participants from several population to find like the data set of like the people, and for the like for different participants. To use the data from same families, we use two family data sets. The first one is Manuel Corpus family. This guy is a scientist. He shared his genomic record and his family genomic record. And also we have another public data set, which is for the UTAF family. We have a big family. They all reveal these data. So we use both of them in order to find a real world data set having family members and also having other people from different population. Now let's start with the first inference attack we have. How we consider that? What's the model we use? So here the idea is we consider adversary, okay? And this adversary send a query to a dataset owner, and this dataset owner just sent back the query result, and this query result is in a privacy preserving way. It's he used differential privacy to to preserve the privacy of this query result. Now this adversary has some information. Okay, how many users are in the data set? Okay, he can find it from public sources. Okay, sometimes it's the metadata of the data set. You can know that how many participants we have. Usually they share this information in public. Again, this adversary, we assume that adversary could know the family members, the existence of family members inside this data set, from genomic dataset, public genomic dataset, from social networking sites or so. And these are real attacks happened actually. From all of this information, he could apply Mendel law. I will show later in details how the adversary can do that. And he can infer the genomic data of one target from this family. How our algorithms work in this attack. So again, the adversary send or generate the query. The adversary is trying to include the members of the same family in the query. For example, he can generate the query for based on age, based on location. Can say, okay, give me the sum of this SNP in the area of Zurich, for example. So he could know, okay, the family is from Zurich, then I'm sure that the family members will be included in the query result. After that, he will receive the results, right? He will receive like sum. So this sum contains or includes the value of a SNP for that guy, for that target, the value of SNPs for other people participating in the query, right? And the added noise we have because of the Laplace perturbation mechanism. Now he can estimate, okay, then he can say, okay, then the sum is like in our paper, we have like, we derive the probabilistic dependence for having the sum. And then from the value, he can estimate what are the values I have for that target and his family. And we prove this equation in our paper. You can find more details. Now, next, okay, I got that sum. How to use that sum? For example, I get the sum of 6. What to do with that sum? So the adversary can use the coin change. The coin change gives the result. For example, if the sum is 6 and we have 4 participants, so we can, okay, we can say like how many people with 0 values, how many people with 1 values, and how many people with 2 values, like the SNP itself. So this is the coin exchange. According to the number of participants, he can exclude any coin exchange for more than the number of participants. For example, here, if we assume, okay, we have 1 participant with 0, 3 with 1, and 1 with 2, here we have it for 5 participants. In fact, we have only 4 included in this query. So he can exclude the third example we have here. He can only accept the first 2. All right, so we have now the permutation. We have the right permutation that we can use. How to find the partitions for this permutation, then to check the validity of each of these permutation, the adversary can use Mendel's law. Okay, so for example, now we got 6. So this 6 can we can make it in a partitions of 1, 2, 2, and 1. So if we consider that we have the father, mother, son, and son. All right. So again, the other 6, we can make it 1, 2, 1, 0. So now we want to check the validity. So we go to the Mendel law. Okay. So if the father is 1 and the mother is 2, can the son be 2? Yes, of course. Can the son be 1? Yes, of course. It's fine. So we can accept this partition. But if, for example, the father and mother, 1 and 2, from Mendel law, it's impossible in the normal, I mean, not if we have any mutation or so. In the normal way, we will never have like the son having the 0 value. So it has to be only 1 or 2. That's why we can like reject this partition. And according to here, if we have different correct partitions, we choose, okay, if our target is the first son, so we can, from again, Mendel law, calculate the probabilities that the son have 2. And according to the highest probability, we can say, okay, then we are sure that this target have the value 2. And in that case, we reveal his genetic record. We reveal the SNP value of this target. So the key results, I mean, after that, of course, we quantify the attack success using 2 metrics. We have a privacy matrix according to the estimation error. Like we try to find the correctness of the adversary. Can he find the correct genetic record or the SNP values for the target correctly? Also, we can use the leaked information metric. The leaked information, we are counting how many correct SNPs the adversary can infer. Okay, so now the key results we got from having this attack is like we could see that the adversary can infer the correct value of the targeted SNPs by up to 50%. Like he can infer correct 50% of the genetic record of a target. What we try to do in our paper, in this work, we try to mitigate this by using the attack information, by using the empirical experimental results we have. So empirically, we try to calculate what's the suitable epsilon value to use, how much noise we need to add. for handling this issue, for mitigating this attack. And according to our result, again, we could achieve 50% better privacy guarantees. We could handle, we could mitigate what we have in our attack. Now, you can find all of these details in our paper, more cases, all the experiments we have, all details about mitigation, and it's also open source. You can find our code here. Next, we are trying to consider more complicated, more real-world queries like math, chi-square, not only sum, not only count, because usually, as I mentioned, for the GWAS, the output of the GWAS will be the chi-square, the p-value, and according to these results, we could know the association between the SNP and the phenotype of interest. Okay, so here again, what's the threat model? And how can we use it for a more complicated attack? So again, here we consider that we have adversary, and that adversary want to perform attribute inference attack. This adversary know that, okay, we have family members, we have relationship between participants in this data set, and he know, for example, like, okay, like the number of participants, because again, it's public data, it can be published or shared with the metadata of the genomic dataset, like 1,000 genomes project datasets. Okay, after having this information, he will ask a query. He will send a query. Give me the math for this SNP. Give me the chi-square for this SNP. He will get the result. These results are differentially private. He will follow the same way our algorithm, the one I showed, using the coordinate change, using the equations we have. And here again, we have new equations like probabilistic equations for the chi-square, for the math. And then again, after that, he can infer the target's SNP. Okay, shall we stop until here? No. You can consider this dataset to be just genomic dataset. Maybe it's not, okay, it's sensitive, but it's not high sensitive. Let's say a dataset for the height, for the color of eye, for any phenotype. Now he will use this data, the genomic record that he infer from this dataset, to perform membership inference attack over a highly sensitive dataset, which include like sensitive disease, like cancer, Alzheimer, and so. So the adversary used these inferred SNPs to again perform the membership attack to know if that participant as a member inside another data set. He wants to know, okay, I know your data, I know your DNA, but still, I don't know if you have this disease. I don't know if you have a predisposition to this disease. So if I can make attack into another sensitive data set, and I could know that you are in the cases, then I got what I want. I could know if you are a member, if you have this disease. And this is what happens again. Usually you can find the population math released publicly, and also sometimes they release the control math because, okay, they said the control are healthy, then we can release some statistics about them. So using these two and sending queries asking about the math, the adversary could know if that target is in the case a group or not. So again, he can use the attribute inference attack algorithm I just explained. And for the membership inference attack, again, the adversary can, he know the math values in the control groups, he know the population math value, he sent the math query, he got the noisy math query, and then using the log likelihood ratio, having two hypotheses, he can accept one of the hypotheses. Like if that guy, if that target as a member or not a member according to the result of the LLR. Now, what are the key results we have in this paper? For the math query, we could, like the adversary can reveal up to 50%. For the chi-square query, the adversary can reveal up to 40% more sensitive information compared to the standard differential privacy mechanisms. Now, for the membership attack itself, the adversary can significantly, like the power of inferring the membership of target can reach 100%. We accept that the member is inside the data set if the power is more than 80%. So after sending almost 100 query for different SNPs, the adversary can be 100% sure that this guy is or this target is in the case a group. More results, more experiments, more about the algorithms we have, you can find in this paper, you can read it, and also it's open source. Now, to mitigate, the other way we have to mitigate, as I mentioned here, what we call the selective hiding model. In this model, we are trying not to increase the noise, not to play with the value of privacy budget. No, we have another technique, another model. What's the technique we follow here? Okay, we have dataset and we have the dataset owner who collects the data and who collect the queries. All right, so if the family members want to participate in this dataset, so before adding them to the dataset, we selectively hide some SNPs. We go and see which SNPs can reveal more sensitive information, which SNPs participate more to find the kin coefficient between the family members. So we hide these SNPs in a way that we still have a good accuracy, good utility for the data set. Now, after that, if the adversary query, for example, the sum of the target and other participants, the dataset owner will send the query over or will calculate the query over this dataset after hiding these SNPs, and he will return back like the response after applying again differential privacy. So here we are following only the standard differential privacy, and after that he sends it back to the adversary, and in that way we are trying to mitigate inferring the value of any target. It is saved, it is private. The adversary cannot infer any more sensitive data than expected. Okay, so in our model, so we are what we try to do for the selective hiding algorithm. For example, if here we have our target is the scientist, the Manuel Corpus in the family of Manuel Corpus dataset, and then he joined the dataset. After that, his father joined. Then all of these like colored positions or areas, they donate the they denote the available SNP positions that can be hidden. So we can hide any of them, but to make our algorithm more to increase the privacy guarantees we have, after, for example, the mother joined, we try to hide the overlapping SNPs. So in that way, we are like increasing the privacy guarantees we have. And also, while hiding, again, we consider having a good utility guarantees. We still want to do balance, a trade-off between both of them. We still need to use the data in a good way. The key results by using this, we are near optimal, the standard differential privacy. Like we are giving the same privacy guarantees, but it's not affecting the utility like the other state-of-the-art schemes. No, we still have a good utility, higher utility than them if we compare it with the privacy we have. More details, more experiments, more about our algorithms can be found in our paper. It's currently on arXiv, and also it's open source. You can find it in our GitHub projects. Okay, to sum up, what are the full model? What we are trying to do is like that. We are trying to find the vulnerabilities of applying differential privacy for genomic dataset by performing attribute inference attacks. We use these data, like the results of attribute inference attacks, what we infer, we use it for another membership inference attacks to find the membership of a target if he or she and the case group, and we are trying to mitigate these two attacks using two techniques or mechanisms. The first one is adding more, like using the Laplace perturbation mechanism. We are adding a little bit more noise, suitable noise. We are computing this noise according to the family members we have in the data set. The other one by selectively hiding the SNPs values for the family before applying the differential privacy. So this is the full model we are working on. As I mentioned, you can read all of our papers regarding this to get more information and understand our algorithms better. So that's all from me. I will be happy to answer any question if you have. Okay, then. Thank you so much for attending, and I hope you enjoyed the lecture.
I don't see questions on YouTube. Do we have questions from the students? All right, that was great. Thanks, Noor, and thanks everyone for watching. So if you have a question, feel free to post it in YouTube video. I think Noor will make her effort to answer it.
Sure.
See you next week. Enjoy the rest of the day. Bye-bye.
Bye.